Okay, hi everyone. Good morning. I'm excited that you could join us for this webinar this morning. I am Allison Footman. I lead the STI advocacy program at AVAC, and today we will focus on an update on the STI R&D pipeline and investments with a specific focus on gonorrhea. So why do we care about gonorrhea? Well, the WHO estimates more than 374 million cases of new sexually transmitted infections were acquired in 2020, including trichomoniasis, chlamydia, gonorrhea, and syphilis. More than 82 million of those were gonorrhea cases. We know that gonorrhea has developed resistance to nearly all antibiotics used for treatment. And we know that new therapeutics are under development, but there are concerns that gonorrhea could develop resistance to those new treatments as well. We also know that when left untreated, these infections can lead to severe sexual and reproductive health consequences, including pelvic inflammatory disease and infertility. And while vaccines to prevent gonorrhea are under development, diagnostics are just as important in detecting and treating these infections. We know that diagnostics currently exist to detect gonorrhea, but they can be expensive, difficult to implement, and there are long times between testing and results and treatment, which can lead to undertreatment of these infections. Syndromic management is used in many countries with limited testing availability. It is affordable. It can lead to the under and over treatment of these infections and the overuse of antibiotics. All of this highlights the need for affordable, accessible, and rapid diagnostics to detect and treat infections to prevent transmission. And then additionally, the WHO has published their global health sector strategies on HIV, viral hepatitis, and sexually transmitted infections for 2022 to 2030. This document provides strategies on ending these infections or epidemics by 2030. It includes several objectives for STI testing, including the importance to support the development and evaluation of new diagnostics and testing strategies for STIs, the importance of screening priority populations for asymptomatic infections, and the need to enhance laboratory capacity. Today, we will have Cécile Ventola from Impact Global Health, and she will review investments that support the development of STI diagnostics. We will also have Birgitta Gleeson from FIND, who will discuss STI diagnostics and FIND's involvement in the development of a novel gonorrhea lateral flow assay. And then we will close with Mandisa Mdingi from South Africa, who will discuss the potential impact of a novel gonorrhea lateral flow assay in South Africa. And so again, I just thank you for joining this webinar today. If you want to learn more about AVAC's work in this space, you can always visit stiwatch.org. Additionally, this webinar will be recorded and the recording will be made available soon. And if you have questions for our speakers, please feel free to put it in the chat or the Q&A, and we will try to get through all of these at the end of the presentations. And with that, I hand things over to Cécile.
Thank you, Allison. Share my screen. Right. Hi, everyone. So I'm here with Impact Global Health, and we were previously known as Polysecures Research. I'm going to be talking about the R&D funding landscape for STIs and specifically for diagnostics based on the latest data from the GFinder survey. The GFinder survey is an annual tracker of R&D investment for global health. We are data mining and surveying funders, developers, clinical trial databases, for example, in 3 different global health areas: neglected diseases, emerging infectious diseases, and sexual and reproductive health. And we do this with an LMIC lens, which means we are looking at R&D that would meet the needs of populations living in low and middle income countries. When it comes to SRH, we have been collecting data systematically since 2018 on the following conditions that you see here, so contraception, STIs, multipurpose technologies, HPV, preeclampsia, postpartum hemorrhage. And this year we've added some new gynecological and mental health conditions and abortion. When it comes to the scope of our SRH survey, it's a little bit different in terms of LMIC lens, because we're not looking at R&D specifically for LMIC context, but R&D that would be LMIC applicable. We're doing that because there is a dual market when it comes to SRH. That's not the case for neglected diseases, for example, but there are some needs also in high-income countries, which represent the commercial incentive for R&D. So we are looking at R&D that is not developed for LMIC context, but could be applicable to this context as well. So for STIs, what are the unmet needs? We need new drugs and biologics. For some STIs, there's no cure available, and for others, we have the problem of AMR, which will require new therapeutics. We also need vaccines, and for all of this, that's the case in both high-income countries and low and middle-income countries. When it comes to diagnostics, though, there is a difference in terms of context. There's no unmet needs for diagnostics in high-income countries because laboratory facilities are accessible most of the time. But for low-resource contexts, it's very important that we have point-of-care diagnostics that allow for easy, affordable, and quick diagnostics of these diseases. This difference with the dual market between medicines, vaccines, and diagnostics, on the other hand, is that definitely has an impact on the level of funding, as we will see, which is why I took a bit of time to explain that. Before we dive into important resources, so we have our GFinder data portal with all our data available for anyone who wants to use it. And I'm going to present from the latest survey, so for this year. And this data will be available by the end of the month in the portal. We also publish specific reports. So the last one we published for SRH was in 2023. It's linked here, and the next one will be published in Q2 of 2025. So what does global funding for STIs look like? You have the data here for each year broken down by SRH condition. The funding for STIs is the dark brown or red line. So the immediate thing that we see is that this funding has been increasing and has been consistently the most funded SRH condition or SRH area since 2020. So we've now reached 280 million for 2023, which is a 29% increase since the last report. That's not the case, for example, for contraception, which has been decreasing, or for maternal health condition like preeclampsia, which are also been funding has also been increasing, but is still not in the same ballpark as STIs. So it looks like STI funding is faring pretty well within SRH. The increase was mostly driven by the industry since the last report, with $50 additional million. But I have to say 20 million of that are actually like a cohort effect, like a new participant to the survey that was not participating before. So it's not an actual increase. But we still have 30 million that are an actual increase from participants who have been sharing their data for years and are increasing their investments. Another increase is coming from the Gates Foundation with 8 additional million since the last report, specifically for STIs. So this is an encouraging trend with funding consistently increasing throughout the years. But what does it look like specifically for diagnostics? So on this slide, you have for each year the breakdown between the different product categories, and diagnostics are in bright orange at the bottom. And so what you see here is that the level of funding for diagnostic has been decreasing, both in absolute and relative terms. So it's gone from almost a quarter of all STI funding to 7% in 2023. And it's also from 2023, it's now at $15 million, but it represents almost a $7 million loss since the beginning of data collection in 2018, and also over $1 million loss since the last report. So that means that while funding for STI has been increasing, funding for diagnostic has been decreasing, and it's mostly been redirected for vaccine, as that you can see here in pink, either for gonorrhea or herpes, and also biologics in brown for the two same conditions. That's not a very encouraging trend for diagnostic funding, but that sort of highlights the difference also in terms of commercial incentives. Like vaccines and biologics will be needed in high-income countries as well, while diagnostics are less needed there. But then when we look at the quality of the funding, what is actually being researched, is a little bit more optimistic, because it looks like most of the research conducted for diagnostics is actually LMIC appropriate. So on the first graph here, you have which conditions are targeted by the diagnostics developed. As you can see, a large part of it is for multiple STIs, which is very appropriate to low resource contexts where several things could be tested at the same time. You also have a huge part for gonorrhea, and these tests often imply testing for antimicrobial resistance. On the second graph, you have the changes between the last report and this year in terms of conditions, and you can see that this multiple STIs is increasing a lot, which is an encouraging sign. Syphilis is also increased a little bit, and for syphilis, we actually only include diagnostics for neonatal syphilis, so it's also like appropriate to LMIC context. Also, when looking at the quality or like qualitatively as what projects are being funded, they are overwhelmingly point-of-care tests, often involving self-collection or mobile apps to help with interpretation of results. So this is also like highly appropriate for LMIC. So this is more of an encouraging trend. The funding has been decreasing, but what is being funded is actually appropriate for LMIC. Another way of looking at the data is looking at funders. So here you have a breakdown between 4 categories of funders: philanthropic, private sector, and public sector, either governments or multilaterals. One important thing to say about funders is that actually almost 90% of the funding comes from only 4 funders. So for the philanthropic sector, it's mostly the Gates Foundation. For the public sector, governments is mostly the US NIH. For multilateral, it's actually only one stakeholder, CARBEX, and for industry, also only one stakeholder. So that makes the sector very vulnerable to any changes when it comes to these 4 stakeholders, whether they would withdraw funds or just decrease. That would drastically change the landscape and the probability for this product to actually come to market. So it's not a great, it's not a very strong ecosystem for diagnostic development. Something that is also worth noting is that the private investment is quite limited. It's only 43% of funding, while it is 95% for biologics and 62% for vaccines. So as you can see, again, this difference in like dual market or not. And that might be a problem also to bring diagnostics up until the end of the clinical development pathway. The majority of public and philanthropic fundings focus on early development diagnostics, and it's often in the later phases that industry involvement is needed. So in the future, it would be quite important to secure some industry involvement to make sure that this product actually makes it to the market. So at the conclusion, while the global funding for STI R&D has been increasing, it's been mostly for vaccines and biologics funded by the industry. For diagnostics, the good news is that most of the research is actually LMIC appropriate, with diagnostic developing point-of-care testing and often for multiple infections. However, the funding has been decreasing over the years, and it also relies on only 4 funders, which makes it particularly vulnerable and shows also the limits of dual market for diagnostics compared to other product categories. In the future, it will be there's a need for sustainable funding, so making sure that this funding is less vulnerable than it is right now, and also partnerships with industry, so to help these products progress along the clinical development pathway and actually make it to the market and to the populations who need them. That is all on my side. Thank you very much.
Thank you so much, Cécile, for a great presentation. I do want to remind everyone, if you have questions for Cécile, please put them in the chat or in the Q&A box. And now we will move to Birgitta Gleeson from Fine. So please feel free to share your screen and present.
Can you see my screen okay, everybody?
Yes, we can. Your slides look perfect.
Thank you.
Thanks. It's a real pleasure to be presenting today on behalf of FIND and on behalf of the AMR team. I've been asked today to present on point of care diagnostics for sexually transmitted diseases. I'm going to focus on two main topics, one being the lateral flow assay that we have developed here in FIND, and then I'm going to touch a little bit also on in relation to molecular diagnostics for gonorrhea and chlamydia. Before I do so, I just want to give one background slide in relation to FIND for people who don't know what FIND is. FIND is a global not-for-profit organization. We're headquartered in Geneva, but we have offices throughout the world in South Africa and India and Kenya. And the main aim of FIND is really to ensure equitable access to quality diagnosis around the world. And we have a particular focus on low and middle income countries. So FIND was established in 2003 as a product development and delivery partnership. The main idea behind this is really to bring together countries and communities, decision makers, and also test developers to try and drive diagnostic innovation, to try and make testing really an integral part of resilient health systems. And we started out 20 years ago with a main focus on TB, but since then we've moved into other disease areas, including AMR, which is a team that I work for, but we also work in areas such as neglected tropical diseases and pandemic threats. I'm not going to go into too much information about FIND, but please, if you are interested in what we do on a day-to-day basis, please do go see our website. As mentioned, I'm going to focus now on the lateral flow assay that we have developed over the last number of years for the identification of gonorrhea. So this is very much a background slide. This shows the progression of the study from when it started in 2018 until the current day. So back in 2018, we conducted a landscape review of diagnostics that were available for both gonorrhea and chlamydia at the point of care. As you know, and as Alison quite rightly pointed out, at the moment, both gonorrhea and chlamydia are being diagnosed based on symptoms alone, which can lead to the over-treatment with antibiotics, but also the under-treatment in the case of asymptomatic patients. So we established that there was a need for point of care diagnostics in low and middle income countries. So out of this landscape review, we held an expert meeting with global stakeholders, which included FIND, WHO and GARP, where we established the need and also built the foundations for the development of what we call a target product profile. A target product profile is really a specifications document. It outlines the characteristics that we would like a test to have. So it will outline things like the sensitivity and specificity that a test needs to have in order to have a clinical impact. Following on from developing the target product profile, FIND then put out what's called a request for proposal. This is really a competitive funding call for diagnostic test developers to develop a test in line with the target product profile that we had published. We then partnered with an organization in the USA called DCN. DCN are a contract research organization who focus on the development of diagnostics, but have a particular focus on lateral flow assays. And we have been partnering with DCN since then. We started developing the test in 2019, and then throughout 2020, we moved through the different stages of the development phase. And then towards the end of 2020, the beginning of 2021, we started seeing some positive results with our biobanked clinical samples. This then allowed us to move into our first pilot clinical study, which was conducted in the University of Alabama. Again, we saw positive results from this study. So we then transitioned the lateral flow to this design freeze phase, which means that the test was performing as we wanted it to, and as outlined in our target product profile, and it no longer needed any further development. This then allowed us to go into our first large clinical evaluation. This was a field study in South Africa, which focused on symptomatic males and females. And that was conducted in partnership with the Foundation for Professional Development. Subsequently to that, we also conducted another large clinical evaluation on asymptomatic patients, and also a study in antenatal care in Zimbabwe. And again, this is with our partners BRTI and the Foundation for Professional Development. And I'm sure Mandisa, who has worked very closely on these studies, will talk about this a little bit more in her talk. So today, where we are is we have a design frozen lateral flow assay that has performed well in the field. This test is now undergoing a technology transfer. This means the test is being transferred from our partner DCN to a manufacturing partner who will then bring the test to market. And we hope that in sometime in 2026, we will have a product on the market. This is basically just to say that developing a lateral flow assay is not an easy process. It does take a number of years, and it does come with some cost. I'm now going to take a step back and I'm going to talk a little bit about lateral flow design and the development of the defined lateral flow assay, and talk about some of the characteristics that we've managed to achieve. So for those of you who don't know, a lateral flow assay is composed primarily of this nitrocellulose membrane. And also it consists of a conjugate pad and a wick pad. The conjugate pad is really where you add the sample. The sample then flows from left to right on this image along the absorbent nitrocellular pad. If gonorrhea is present in the sample, it will bind to this gonorrhea test line, which is coated with antibodies that are specific for an outer membrane protein of Neisseria gonorrhea. And this is then bound to a secondary antibody, which is conjugated with the fluorescent probe, which allows visualization of the test line if gonorrhea is present. That's to say, actually, this test requires reading with the fluorescent reader. It can't be visualized with the naked eye. The nitrocellulose membrane also contains this control line, which is an internal control, which must be present if the test is performed correctly. The sample then continues to flow along this membrane, where it's then collected by this wick pad at the end of the assay. So this is to say that FIND has developed a 20-minute, really easy to use point of care assay for the detection of gonorrhea. So far, the test has only been validated in female vaginal swabs and male urine. However, we do hope to incorporate additional samples in the future. As mentioned, our target is an outer membrane protein of Neisseria gonorrhea. We've managed to achieve a relatively good analytical sensitivity. Obviously, our analytical sensitivity is not as high as you would see with molecular tests, but we have managed to achieve a limit of detection of about 1 × 10⁻³ to 5 × 10⁻³ colony-forming units per mil. However, this is dependent on the reference strain of gonorrhea that you are testing. And we have shown that we are able to detect 31 various different NG strains that have been collected globally. Again, as I mentioned, with a varying limit of detection. In terms of cross-reactivity, we really also wanted to show that the test doesn't cross-react with other commensal pathogens. We've shown no cross-reactivity with any of those pathogens. However, we do see some cross-reactivity with other Neisseria species. So we do see cross-reactivity with Neisseria meningitidis, Neisseria lactamica, and Neisseria polysacchara. This is not so much of an issue when you're trying to test genital samples. However, it does become a bit of an issue if you want to test with, let's say, pharyngeal samples where there is commensal Neisseria meningitidis. This next slide really shows how easy this test is to use. It's very similar to COVID lateral flow assays that people are very familiar with. This here is with a vaginal swab. You take a high vaginal swab, you then put the swab into the buffer, you then twirl the swab approximately 10 times and then leave it within the buffer for about 1 minute. The main idea behind this is that you really want to extract all of the cells from that matrix to be able to expose that antigen so that the antibody can bind if gonorrhea is present. You then twirl the swab again for 10 times to ensure efficient extraction of the cells, and then you then drop 4 drops of the extracted sample to the sample port on the lateral flow cassette. You then allow the sample to run along that nitrocellulose membrane for about 20 minutes before then inserting the cassette into this fluorescent reader. So this fluorescent reader is a small, light, portable, easy to use reader that's operated with an external battery pack similar that you would use to charge your mobile, and it's designed to be used at the point of care. Then after about 30 seconds-ish, you get a result from this fluorescent reader, and it lights up letting you know if the test is positive, negative, or invalid. In terms of its performance in clinical studies, our first clinical evaluation, as I mentioned, was in symptomatic patients in South Africa. We managed to show high sensitivity and specificity, which was in line with the requirements that were set out in that original target product profile that was published in 2019. We show that in symptomatic men, the test has a sensitivity of 96% and a specificity of 97%, which did drop slightly in symptomatic women, where we see a sensitivity of 92% and a specificity of 96%. Turnaround time for this test is less than 30 minutes. It's about 25 minutes if you include sample collection, which is a lot quicker than some of the tests that are currently available, in particular the molecular tests, which can take up to 90 minutes for time to result. 2 following clinical evaluations took place. One was a clinical evaluation among asymptomatic males and females, and this was in South Africa. We did see a drop in our sensitivity and specificity in our asymptomatic population. This was as expected. Our sensitivity dropped to 81% in asymptomatic men. However, our specificity did remain quite high at 94%. In asymptomatic women, contrary to what we saw in the symptomatic study, we did see a slightly increased sensitivity and specificity in women in comparison to men. Following on from that, we conducted a study in Zimbabwe, and this was among patients in antenatal care. This study was in partnership with BRTI. We did see quite a reduced sensitivity with the test in this ANC population. We're not 100% sure why we're seeing this drop in sensitivity. There has been some hypothesis that it may have something to do with physiological changes that happen as a result of pregnancy, which is having some impact on bacterial load. So as a result of this test, we are now planning to conduct 2 additional studies looking at the ANC population in particular. So what's next for the lateral flow assay? Obviously, we're seeing a test that performs well. As I mentioned, we're now undergoing a technology transfer with ongoing negotiations with potential manufacturers. We're also working on a regulatory pathway and a go-to-market strategy to ensure that when the test is ready to go on the market, that this is streamlined. We're also gathering more evidence in relation to the lateral flow assay. We're looking at other geographies and other populations and subgroups to make sure that the test still performs just as well as in these additional populations. We're also now beginning to pilot for self-collection, and this again will be a study happening in South Africa, and pilot for extragenital samples, which will be a study that's happening in Papua New Guinea. Throughout all of these studies, we always look at the usability and acceptability of these devices to ensure that they are easy to use and acceptable in routine care. In parallel to that, we're also modelling the impact of the introduction of these lateral flow assays, looking at things like its impact on transmission, but also on the development of antimicrobial resistance due to the decrease in the use of antibiotics associated with the introduction of a diagnostic test. And I'm sure Mandisa will talk about this a little bit more. And also looking at the cost effectiveness of introducing a lateral flow assay. As you know, a lateral flow assay is a lot more affordable than some of the molecular tests that are currently available. When we started out this project in 2018, it was our ultimate goal to have both a test for gonorrhea and chlamydia. The development of a test for chlamydia is a lot more challenging, but over the last couple of years, we've partnered with the US CDC, funded us, and we've partnered with the University of Southern California to work on developing a lateral flow assay for chlamydia, with the ultimate goal that we would have a dual NG-CT test. But as I mentioned, it is challenging, but it's something that we're currently working on. Next, I'm going to touch a little bit on molecular tests. As you know, as a result of COVID, there was a lot of innovation in terms of diagnostics. Diagnostics became smarter, faster, cheaper, and easier to use, and definitely became more available closer to the patient. When the COVID wave began to recede, a lot of these diagnostic companies have now moved into the development of STI molecular tests, which is great for this space. And obviously, molecular tests, they're a little bit more complex to use than a lateral flow assay, and they're probably not as affordable as a lateral flow assay. However, they do have advantages in terms of increased sensitivity and increased specificity. So based on that, FIND did publish a request for proposal again just a couple of months ago, where we're seeking manufacturers for the development of a true point-of-care molecular test for both gonorrhea and chlamydia, and to try and accelerate product development and evaluation and market entry with a specific focus again on low and middle income countries. So this is quite an exciting space to watch over the next few months. Just to say, any of the tests that I've shown today and the lateral flow assay are obviously available on our FIND test directory, which really shows all pipeline technologies and tests that are available on the market. So please, if you're interested, do have a look on our website. And finally, just to say thank you for everybody who's involved in the study. This work would be impossible if it wasn't for all of our partners at the University of Alabama, the Foundation for Professional Development, and also through ZIM, which is the BRTI group in Zimbabwe. And finally, to thank all of the FIND team, in particular Benjamin Blumel, who leads this project at FIND, but was unable to present here today, and also to our funders. So thank you, and happy to take any questions.
Thank you so much, Birgitta, for that amazing presentation. I know that we will get to some of the questions folks have for you in the chat later during the webinar. And so now we are going to have Mandisa and Mdingi present.
Thank you, Alison. Hi, everyone. My name is Mandisa Mdingi. I am from the Foundation for Professional Development in the Eastern Cape, East London, South Africa. I'm excited to share our work titled A Novel Lateral Flow Assay for Point-of-Care Detection of Neisseria gonorrhea. And my presentation will be focusing mainly on our implementation experiences. I think it's important to share our experiences based on implementation, because this point of care test could actually provide, could actually contribute to bringing, rapid access to diagnostic testing, given the rising importance of STI timely testing, and as well as we are hoping that our work would contribute to broader discussions in enhancing STI testing in our settings and other lower middle income countries. Thank you. Next slide. I would like to acknowledge the FIND team for providing financial support for this study. Other than that, I have no other disclosures to confirm today. Next slide. Thank you, Alison, for sharing that comprehensive information on STI burden, specifically for Neisseria gonorrhea. So now where we are, or I will kick off with looking at why do we need an affordable point-of-care device? Based on the information that we have already shared, focusing on the global burden of STI, specifically with Neisseria gonorrhea, we therefore do need an affordable point-of-care test that could help us to strengthen the management of STIs. And also Birgitta touched on this specific focus on target product profile that was developed for mainly to be used in syndromic management settings, including our setting, South Africa, and as well as screening for high-risk populations where we think of sex workers, pregnant people. And I'm only sharing in my screen the similar slide that Birgitta just shared, looking at the specific requirements for the TPP for these non-molecular test point of care of gonorrhea. Thank you. Next slide. So I will dive into what we did here at FPD. So first of all, as I've mentioned, that this was a study that funded by the FIND team. FIND team came to South Africa late in 2020, conducted a training for higher team, including the managerial staff. And this was followed by training of field team. And they also provided us with the procedure quick cards, and these were to be used in different or for different types of samples to be collected, since we're going to look at different population, that is men and women, which I'll talk more in my next slide. After this training, we then moved to step by step kind of we did where we went to sites and we sat down with the team, and we looked at how to perform the test, in also preparing them for real life situation where they would also get maybe what to do when you get invalid results, what to do when you get positive, how do you confirm that this is a positive result, this is a negative result, and what do you do? So we looked into detail as to preparing the staff for the implementation. This was a cross-sectional study of diagnostic performance of this lateral flow assay, and we compared it to the gold standard GeneXpect assay, CT and NG. So that means we did not treat anyone based with positive results based on the LFA results, but we treated everyone that tested had a positive test results as per GeneXpect. And this study was done in 5 primary healthcare facilities in the BCM area, which is Buffalo City Metropolitan District in East London. And within these 5 facilities, we had in-facility GeneXpect system installed there. And just to mention, the study was approved by University of Pretoria HREC, and all the study participants before participating in the study consented to participate in the study. We included men that presented with male urethral syndrome, and the number for those were 200 men, meaning these were men that presented with symptoms. And for women, they presented with vaginal discharge syndrome. Again, there were 200 women that we included in the study. And again, for the follow-up phase, as Birgitta mentioned, we also recruited asymptomatic men and asymptomatic women. Also, just to mention, part of our study population included the partners with notification slip. However, those numbers were quite less compared to men and women that presented with symptoms. Thank you. Next slide. This slide is meant to show the procedure, how it's done, which Birgitta has touched on it previously, where the staff would start preparing the buffer. And then after collecting the swab, which is collected by, it's a nurse collected swab, they will then prepare the buffer. And then after preparing the buffer, as you can see the next picture, then they will collect they will then get the specimen depending on the type of specimen, but for this purpose, this was during the training phase. So this is just water with the coloring thingy. And then after preparing the sample, they will mix the buffer together with the sample, and then set the timer for that 1 minute while preparing the cassette or the cartridge. And then after that, they will set 20 minutes and wait. At that same time, they can perform other things depending on how busy the site is. And then only after 20 minutes, they will then read the results. As you can see, that small, that small black device is actually the lateral flow assay, where you would then insert the cassette after 20 minutes, then you read the results. So if you can see there, there's a light on the positive sign. It's an indication of a positive result. And for any negative result, it will be on the minus sign. And for the invalid result, it will be on that exclamation mark in between. Thank you. Next slide. Again, Birgitta touched on these results. We really did see high rate. I think it's important to highlight the high prevalence of gonorrhea in our setting, with for men sitting at 64%, and this will be those men that presented with symptoms, and for women they presented or they had prevalence of 18%, which is quite high in our setting. And the testing really did perform well in terms of sensitivity and specificity, already within the TPP requirements. And this is exciting to actually see these results in real life setting, not only in the lab. And this work was published by Professor Remco Peters and colleagues. You can see the title of the paper. And again, if you look at the asymptomatic evaluation of the same population, I mean of the same setting, but just different population with no symptoms. As Birgitta mentioned, the sensitivity for both females and males did drop a bit, and this work is not yet published, but has been submitted to Lancet Infectious Diseases. Thank you. Next slide. As part of our study, we also had the implementation component, where we're interested to look at the usability and acceptability of this method. And then this was done amongst our research nurses and the field workers, which mainly work as research assistants for screening and recruiting of these study participants. And I'm excited to share today that we had our results for usability and acceptability amongst our staff showed high acceptance of these methods. And I think most importantly is to mention that this method can also be used by staff that does not necessarily need to be used by professional nurses, since our field workers were also able to perform the test after being trained. And this implementation work was also published as part of the peer-reviewed articles. Thank you. Next slide. So I'll now talk on the potential impact that this assay could have on clinic flow. Since we evaluated the device in the public healthcare facilities, which I think for every device or medical device that is being evaluated, it should be relevant to be done in that same setting, where at the last stage the device would then be part of the testing at the clinical setting. It's important that we share our experience and say that these tests, as I've mentioned, can actually be performed by any staff members, just provided that are trained to be used, and it can actually reduce or contribute to reduce waiting times, because while one is performing the other part of the test, and the other person can also take history and do the other things. And also it can also accommodate the space constraints and potential stigma concerns, which we know STI in general has got that stigma in our communities, and people don't want to come to the clinic. But now, if they know that the test is just going to take less than 30 minutes, that can also contribute to motivate them to come to the clinic. And also, if you consider the availability of space in our public healthcare facilities, this is a small device that can actually be accommodated on top of the table without necessarily having to occupy a larger space within the consulting room. And also, we observe from our participants as well that the test itself has got a potential to increase awareness in terms of STI types amongst patients, because this is also the gap that we have identified during the implementation of the study, that our communities are not aware of the different types of STIs. And now if you were to bring in this test and then you say this is specifically to test you for Neisseria gonorrhea, and then they now become aware that there's actually this type of STI that can be treated if someone does present to the facility in time. And also, lastly, it also contributes, or it's got a potential to contribute to investment in healthcare, improving technologies, helping us also to minimize the AMR possibility, which we know that in our settings currently we use syndromic management, which has its own pros and cons, being the ability to not being able to monitor the trends of STI per participant. So we believe that if this test does have a potential for the healthcare or the public healthcare sector to improve in terms of STI management. Thank you. Next slide. So what are the next steps? As Birgitta mentioned earlier, we are excited here as FPD team. We are looking at evaluating the device again. We are in the preparation phase of doing a third phase of this of evaluating this device, where we will be looking at the performance of the same device, but now including pregnant women, which were not initially included in the previous population that I've mentioned, as well as looking at the usability and acceptability of self-collected samples, because previously we had used the nurse-collected samples in both the symptomatic and asymptomatic group. This is exciting for us because now this will show if there's really differences in terms of nurse collected and participant collected samples, which also going to contribute to STI management, because if the patient are now equipped to collect the samples themselves, that can also contribute to reducing waiting times in our facilities, especially in that STI setting, where that alone can also contribute to empowering and actually encourage our communities to present to the clinic for STI treatment. In conclusion, this next slide, please. In conclusion, this lateral flow provide a promising point of care test that might strengthen the STI treatment, especially in the syndromic management setting. So as FPD team, we are really excited to be part of this journey with FIND towards improving public health management in our country. Thank you so much. I would like to acknowledge my team that is behind the wonderful work that has been done for the past 2 years, as well as the Province of the Eastern Cape Department of Health and FIND Diagnostic for funding us for this project. Thank you very much.
Thank you so much, Mandisa. And now we will move to our Q&A session. So again, if you have questions for our speakers today, please put them in the chat or the Q&A box. And so Birgitta and Cécile, would you mind coming on camera? Great. And so our first question for today is, Birgitta, what symptoms are you referring to when you refer to symptomatic men and women? Is this vaginal or urethral discharge?
Yes, correct. Both vaginal and urethral discharge.
Yes, for male and female, that would be categorized as symptomatic in our case. We don't look at things like abdominal pain. It's not included as part of our inclusion criteria. So yeah.
And what is the cost of this gonorrhea lateral flow assay?
Yeah, good question. So as I mentioned, the test is just being transferred to a manufacturer, so we don't have a final cost. We would hope, though, that the test is in line with the cost that we've outlined in our target product profile. For a lateral flow assay, I think our optimal price is around $3. So that is the hope. And anybody that we partner with to manufacture this test has to agree to our global access terms. So that is our aim. But let's see what the final price actually is. And that's the best I can do for today.
And Mandisa, so we know that you were able to do some qualitative work, and were you able to collect the views of providers, clients, and policymakers on the adoption of this test? And if you did, what did you find?
Thank you. Good question. Thank you, Alison. As I've mentioned, for the first two phases that we've done for this study, we looked at the experiences for only based on our research staff, included the research nurses and the field workers. So for the planned study now that is going to be looking at evaluating the device in pregnant women and those non-pregnant symptomatic women, the part of that protocol includes now capacity to collect views of both the providers in the clinics and the policymakers as well as the clients, because the clients will be looking at evaluating their views on collecting the samples.
Thank you, Mandisa. And Cécile, you've done such great work in looking at investments and understanding funding data. Is there anything that we as researchers and advocates, policymakers can do to bring more attention to STIs to maybe increase this funding?
I mean, I'm not an expert in advocacy, but I would definitely say that anything that sort of ties up the consequence of not caring more about this can help governments and funders everywhere to understand that, like, even a local project can actually really contribute to the overall situation. So yeah, we're mostly looking at like the funding in the pipeline, how to translate that into advocacy. I mean, of course, like these numbers can be used to show what happens when things are funded, what happens when things are not funded, where are the gaps. That's definitely something that we do with our data. And I think for STIs, yeah, again, there's this like opposition between what is a commercial incentive in high-income countries and what is not, and make sure that there's enough research on things that are maybe not something you can sell in high-income countries, but that would still benefit the population of everyone.
Thank you, Cécile. Another question for Birgitta. So there are FDA-approved point-of-care devices like Visby and Binks that offer high clinical accuracy of molecular results. Is the antigen-based still preferred, mainly for cost considerations?
Yes, exactly. Yeah. So lateral flow assay would be more affordable than
The point of care molecular tests that are currently available. Obviously, like I mentioned in my talk, there is advantages to having point of care molecular tests. I do think this space is opening up to more affordable molecular tests, also, as I mentioned. But yes, a lateral flow assay would be more affordable. That's the idea behind it. It's much simpler to use. And that's the main advantage of having a lateral flow assay. And Mandisa, you can speak to that a little bit more as well. I mean, Mandisa works with this test, so she knows it better than I do.
Mandisa, would you like to add to that?
Yes, definitely. I think also having the experience of worked with GeneXpect before, I think this test would be an affordable one, and also in terms of sample processing time, it's definitely going to bring a change because there's lesser time to reading of the results, and also not just only that, handling as well, being easy to use, and the fact that I think with the capacity that the test health itself, where in our context, sometimes in South Africa, where we are faced with load shedding context. So I think the test does stand a good chance.
And Mandisa, another question for you. So, will you include the poor, the marginalized, and disability cases in future work? Maybe what are some other marginalized populations or key populations that you hope to include in future studies?
I would say at the moment, definitely it would be interesting to look at the other population, the high risk population, for instance, the MSMs. I mean, we have a potential to look at that population again. I mean, as well, that will be interesting to look at.
And Mandisa, in thinking about some of the current syndromic guidelines for STI testing, what do you think that this novel, this lateral flow assay can help in either changing testing guidelines or recommendations? How can we pair these two together?
First answer, I think it will definitely bring change in terms of the number of treatment that the clinician would offer to patients, because then the patient will get that targeted treatment compared to the syndromic management, where they would get a whole lot of antibiotics. And also for if it were to be implemented or to be part of the guidelines, it would improve the STI management, because we know that the syndromic management has got that disadvantage of missing the asymptomatic group. So if this test would be included into the routine screening, that will mean even the people that presents with no symptoms, upon being tested, they will then be picked and then they will be treated. So we won't have the missed test, the missed participants, especially when they have presented to the clinic. So it will provide us with the opportunity to treat the asymptomatic, to reduce the antibiotic burden that would be given in case of syndromic management.
So this next question is for Cécile, but I think all of our speakers might have some thoughts on this as well. And with STI testing moving to point of care, at home, or self-testing, how can we continue to monitor accurate reporting? So the CDC currently does with lab-based testing, but how can we still continue to monitor these cases with our testing mechanisms changing and sometimes being more patient-centered instead of lab-centered?
Yeah, this is really beyond my expertise, but I would assume that with everything that has some sort of independent testing in place, then you can actually look at drug prescriptions whenever that requires the prescription of some treatment. That would be another estimate. But I also don't think that that it should slow down the development of self-tests, because we all know that they are very impactful for a lot of reasons. So maybe we need to be more innovative in monitoring rather than restricting self-test.
And Mandisa, is this a topic that has come up when you've spoken to, healthcare providers or people who are conducting this test, concerns about monitoring results and reporting results as we move to more self-testing?
Yes, some of them would indicate that, as part of, you will remember that for every device that is being implemented, it's important to have that continuous quality control and quality management to ensure that actually the test does perform what is expected for it to give. So this is also a part of what we do or what we have done for the study, doing the quality control, the quality checks, looking at whether the device does give negative results when it's tested with using the negative controls. So quality management is definitely part and parcel of this implementation.
Well, thank you so much for that response. And thank you to all of our presenters today. You guys provided such great context on STI diagnostics and investments and what the future really holds. I think a lot of us are really hopeful and excited for some of these future tests and hopeful that more funding will come down as well. And so with that, this is the end of our webinar. We did record this session. The recording will be made available soon, along with PDF copies of our presenter slides. So thank you everyone for joining us today. And I hope you have a great rest of your Thursday.
Thank you, everyone. Thanks, Alison, for organizing.
Thanks, Alison. Thanks, everyone.